Hi Amrita, can you hear me?
Yes, ma'am, I can. Good
Or
evening.
good evening, hi. We'll wait for a couple of more minutes for people to join. Hopefully, a couple of more join.
Sure, ma'am. So, how has the course been going for everyone so far?
Good.
Okay. Has it been tough, easy? Are there any particular things that that need further explanation? Or you would like to cover something more in detail, or would you like to know something more about certain topics? Is any of you from an ecology background or non-ecology?
Non-ecology.
Non-ecology. With what is your background, Architram?
I'm doing electrical electronics.
Okay, cool. And you are interested in ecology?
Yeah.
All right. Do you do anything, any birding or any observations, hikes, treks, etc.?
I have done, and I am interested in spotting insects, different types of insects.
Okay. That's great. That's great. Awesome. What about others? What are your backgrounds, if not ecology?
I'm a biotech graduate, so I think it's partially in there and partially not.
Okay, yeah, all right. A little bit linked, little bit not, but yeah, good to have you here too. I think we're just about 6 people today, very low turnout. But since Architam mentioned that you like documenting insects, etc., I was wondering if you use the platform known as iNaturalist, or have you heard of it? Has anyone heard of it?
No, I have not.
Okay, so now since we're doing a course in wildlife ecology, let me just introduce myself. So I'm Chiti Arvind, and you're one of your 3 course TAs for this course in wildlife ecology. Welcome to week 5. And so since we're all interested in some way or the other in ecology and want to learn more, there are a lot of citizen science platforms which you can use to document ecology, and one such platform is known as iNaturalist. I will just put this up on the screen. So let me know if this has loaded for you. So this is a platform called iNaturalist. It is I think a part of the National Geographic platform. And what is really cool about iNaturalist is you can document. It's an app that works on your phone, and you can also use a web browser. You can click pictures of all any organism around you. It can be plants and animals, and you can upload it to this portal. You can create a profile, upload it to the portal, and you will get identifications. So it uses an AI to identify these organisms, and if it's not been identified, there are experts that are a part of this platform that will help you identify it. So it's a great way of documenting biodiversity around you or in places that you go visit and you're interested in. So you can follow different pages, and then slowly as you build your expertise in any field, you can also, serve as an expert for a particular taxa. Like for example, after documenting a lot of butterflies, you have become an expert on butterflies, at least in your region. Then when somebody posts a photo, you can give your suggestions as to what you think the possible ID of the species could be. So this is a great citizen science platform that is used all over the world, and you can get like some great data out of this. I know a lot of people do use Google Lens to identify anything around them, but iNaturalist also works as a really nice scientific community. So that is perhaps something people can explore if they are interested in. I'm just going to put the link in the chat so that you can access this. Alright, enough about that. And there is also another platform known as eBird. So if you are interested in birding and documenting birds around you, you can use eBird to create checklists, etc. And if you're also interested in identifying birds, there is this app called Merlin, MERLIN. Both these apps are developed by the Cornell Lab of Ornithology, and it's a huge community of scientists and non-scientists who come together just to document birds all around. So like this, there are perhaps even platforms for several other taxa. Alright, so welcome to week 5, and I hope all of you have gone through your lectures. Today we will be covering a little bit on population ecology, and I hope this can be an interactive session. Everybody please feel free to pitch in, use the chat box or unmute yourself, and let's have some discussions. So when you see this equation on the screen, Pn plus 1 equal to Pn plus natality plus immigration minus mortality minus emigration, what does this actually signify? The bird app is called eBird. I can just and the ID for birds app is Merlin. Okay, so tell me, those of you who can see this on the screen, let's not worry about it being an equation. What does it signify? Any ideas, any suggestions? Nobody knows?
About how much population is there in a particular area?
Yeah, excellent. That's it. You're monitoring population movement, right? I mean, you this is just an equation that shows population dynamics, because there are 4 variables that are shown out here. Yes, you can estimate population size, etc. Could someone give me the definition of natality, immigration, mortality and emigration? Okay, let's start with natality. What is natality? No one knows what's natality? What does it sound natality is? What does it sound like? Birth rate. Excellent. Yeah. And what about immigration?
Influx of any birds or species.
Correct. So movement into a population is known as immigration. What about mortality?
Death rate.
Correct. And emigration?
Outflux.
Exactly. So the equation is really simple, because it shows movement into a population and movement outside a population. So when you add new individuals to a population, they can be added in 2 ways. One is via migration into a population, which is known as immigration, or one is via new individuals being born. That is called natality, right, or birth rate. And the ways in which individuals are eliminated from a population is known as, can be in 2 ways. One is mortality, or the amount of individuals dying out, and another one is efflux from a population, or emigration, right? So your Pn plus 1 is nothing but the population in a particular year, which is just the population in a year. In the current year is known as Pn, and in the next year is known as Pn plus 1. N is nothing but, just let's consider it at this time as the next year. So the population of the next year would be equal to the population of any organism in the present year, or let's say population size in the present year, plus the number of new individuals that are born, plus the number of individuals that are getting into the population, minus the number of individuals that are dying, and minus the number of individuals that leave the population, right? So this is like a very, very simple equation, but it is very, very important, right? So when in terms of just mapping species distribution and mapping the population densities of any organism for conservation practices, you need to know something very crucial, which is the population of this organism, right? Because you need to know how many individuals are there, how far do they range, whether the areas they cover. So if you don't have information on this, such quantitative information, it is very difficult to come up with applied conservation management plans for species that are especially on a decline or that are threatened by any factors. They could be climatic factors, anthropogenic factors, any such thing, right? So population dynamics is a really important topic in wildlife ecology, and you will find population dynamics getting into anything that you're studying, like even if it is something not ecology, if you're looking at evolutionary biology, that comes in. If you're looking at ecology in terms of policy management, that is very important. So yeah, if you do have any additional things that you want to discuss in this topic, please feel free to yeah, let me know. Alright, so a population's dash, you have to fill in the blank with the words that are given, can be defined as the number of individuals per unit area or volume of habitat. Feel free to use the chat box with any answers. Okay, we have one answer in. What do the others think? Okay. Okay. What do the others think? So, okay, so everybody seems to be thinking it's D. Why is it not A? Why is it not a population's geographic range? Any thoughts on why it cannot be geographic range? Or is it geographic range but not density? Any thoughts, anyone? Range is just an area. Okay. Prashanta, would you like to elaborate on anything more? You are on the right track. Okay, sure. Yeah, in classical definition, density is any number of something per unit area or volume or whichever. So, yes, it's definitely not geographic range, because the geographic range just shows you extent where an individual is found or not, where in the global map, an individual a particular species is found. Let's talk about species, where a species is found, but it doesn't tell you how many individuals are there, right? So, what is dispersal pattern? Does anyone know what is dispersal pattern?
It's the extent to which a particular species or a group of animals are dispersed within an environment.
Okay, could you elaborate a little bit more on disperse? What does disperse mean?
Maybe the distance at which they are located.
Sure. And distance from?
One another. Is that
okay. Okay, sure. Yes, but as you rightly said, it's a measure of distance, and distance in terms of movement, right? So most often when people talk about dispersal and migration, any ideas what could be the difference between dispersal and migration?
Is dispersion contained within a geographic boundary, but migration happens from one zone to another?
Yes. So let's get into a little bit of finer details. Dispersal is, as you mentioned as well, when you talk about the scale of the events, migration happens usually over a larger scale. Dispersal happens over mostly shorter scale, but you can have dispersal over larger scales also. But what is the main difference, or like the most stark difference between dispersal and migration? Any ideas? Okay, so like dispersal is movement. Okay, movement, let's say for example, we usually talk about dispersal in terms of dispersal from a natal site or a natal territory. Natal territory, what do you think natal territory means? We covered the word natality in the previous slide. Yeah, go ahead. Anyone?
Native background, native area.
Yeah, what does native area mean? Natal area or native area? Well, for humans it's a little bit skewed, but what does a natal territory mean?
Where most of the species are concentrated.
Concentrated. What does natality mean? What is the definition of natality?
Birth rate.
Birth rate, right. And what does natal mean? It's all in the word.
Is it the place where the organism was born and
Correct.
it usually inhabits that zone?
Yes, absolutely. So you have two words, natality and mortality. Natal place is somewhere where an organism is born. Okay, so any organism is born, let's say for example an animal that moves is born in a particular place, but dispersal from its natal territory is something what is usually measured in ecology. So the amount or how far an individual moves from its natal territory to set up a territory of its own, let's say for example. So that covers something on natality. What is migration? What do you think when you think of migration? What's the first thing that comes to your mind?
It could be seasonal.
Correct, right? It's seasonal. And what does seasonality in migration mean? You're right. You're on the correct path. So when you say something is seasonal, what does it mean? Let's just break it down. So when it's seasonal, it means that in some season it does something, and in another season it does something else, right? In terms of change. Does anyone agree with me or not?
Yes, ma'am.
Yeah. So seasonality just means that there is a recurring event, right? So when you say migration is seasonal, it means that migration is a recurring event, right? So if you talk about I don't know, like humpback whales, or you talk about monarch butterflies, or you talk about some albatrosses, these are like the classical examples of migrating organisms. At certain points of the year they move somewhere, and at certain points of the year they come back, right? So it's a continuous movement that happens during the act of migration. But during dispersal, what usually happens, it's a one-way movement. The organism is born in one place, for example, and it moves to another place. It doesn't return back to the place where it is born, right? Especially for non-social organisms, they for example, let's say, I don't know, let's say some lion, or lions leave the males leave their natal territory and join other prides, right, in another area to set up their own territory. They don't come back to their original natal territory where they were born, right? So that would be considered as a dispersal event. So that's the main difference between dispersal and migration. One is a one-way process, and one is a recurring process that cyclically happens. Alright, so here we can see a map, and could anyone take a guess as to what this could be? I expect multiple answers for this. Multiple people answering for this. Feel free to use the chat box. What do you think this figure represents? Should I
Population density.
Population density. Okay. Population density is correct. Population density of You can all take guesses on the chat. I'm going to wait for a couple of guesses. Nobody? Nobody has any guesses? How's that possible? No one can guess what the species could be? You can throw in any random guess and then we can discuss. No one? No replies? Okay, do you think it is a vertebrate or an invertebrate? Does everyone know what is a vertebrate and invertebrate? Yes, no? You can just say yes or no in the chat. We can even discuss what's a vertebrate and an invertebrate. Can someone tell me what is a vertebrate and an invertebrate? No one? Really? Okay. Can people who know what a vertebrate is put their thumbs up? Give a thumbs up. Okay, thank you. One person knows, two people know. Three people know. Four. Okay, so some of you at least know that what is a vertebrate and invertebrate. Could you tell me if this particular distribution that you see is and belongs to a vertebrate or an invertebrate? Take a guess. Or is it a microorganism? No guesses?
Vertebrate.
Okay, excellent. Yes, indeed, it is a vertebrate. Can you guess which vertebrate this is? Is it a reptile? Is it a mammal? Is it an amphibian? Is it a fish?
Mammals.
It's a mammal. Okay, that we have shortlisted. No, it's not a bird. Okay, so which mammal do you think is distributed like this? White is zero distribution, and darker you go in red, that is more distribution.
Humans.
Yeah, exactly, right? Amrita says primate. Okay, close enough. But yes, this is a density map of human distributions throughout the world, right? What is the biggest giveaway for this? What is the highlight? What stands out?
A very high population density in India.
Right. So India and China have a super high population density. Can people tell me, can you see my mouse? I don't know on the screen. Is it visible? Probably not. No? Is it visible like a mouse? Yes, no? I'm using a pointer now. Can someone, okay, I can't read the chat. Okay, someone said yes. Okay, so
Yes,
yeah, all right. So these areas which have lighter colors on the top, out here, why do these have very less density of humans?
Because of lack in resources.
Lack of resources of, could you elaborate a little more?
Maybe water.
Okay, what do these areas mostly comprise? You're correct. You're correct about that.
Because sunlight is more on those areas, that's why water is less.
Okay, so the areas in the center, probably out here, the sunlight is more. But towards like the poles, the sunlight is very less. But the first thing that you said, like availability of water, right? That is most important for any living organism, especially humans. All these areas that are very light in color are mostly desert, right? They are cold deserts or sandy deserts, barren land, uninhabitable. So this is literally one of the few species that has managed to colonize every single landmass, right? You find humans everywhere. They have found some way or the other to occupy some niche and created modifications around their environment to be able to survive, right? So population distribution and population density estimation are important factors. So if you see a species distribution map of any other species, you would like species as in like vertebrate species, you would not see it extending to such a large area, right? So yeah, this is just the scale of human inhabitants. Even I can't see, it's like too small, but these are about very few individuals to probably, like, the last goes to couple of million individuals, right? So yes, this is a population distribution map for human beings. Okay, we already discussed most of this, but I would like to hear from all of you as well. Why is it so important to assess population density? We just discussed this. Why is it important?
Maybe you could give us an idea of what kind of regions are better preferred by that particular organism if their population density is quite high in those areas.
Right, that's correct. That's correct.
Maybe to see how much resources are there in that particular area.
That's interesting. You can use, that's a really interesting point. You can use populations of certain organisms to track resources, or use them as a proxy to track resources. If there are any associations between particular organisms and particular resources, you can definitely do that. Yeah, sure. What do you think can cause changes in populations?
Temperature, the humidity, or the availability of water.
Yeah, environmental factors. We can broadly categorize all these things as environmental factors. Yes. What are the other things that can cause fluctuations in population density of any animal or plant? Let's say any organism.
Predators.
Sure, yeah. Biotic factors such as predators, definitely. Immigration, natality, mortality, disease. Yeah, excellent. Disease. There can suddenly be an outbreak of disease somewhere, completely unpredicted. Suddenly, none of the individuals or species are able to cope up with any situation or any infection, and then there is a huge loss in population numbers, right? And when populations drop to very low numbers, why are they at risk? Why do people start worrying when population number drops of anything, or start dwindling, or are under threat?
They may indirectly affect the environment.
Okay, could you elaborate a little more?
Like they will affect the food chain.
Yeah, that's an important point. Yeah, they'll affect the food chain. There'll be a disruption in the trophic level. So, so when we are trying to, assess any particular organism in its environment, what is it important to know? What is important to know? Like, let's just take for example conservation of tigers. Why should tigers be conserved, or why should elephants be conserved? No idea? So each organism has some role that they play in the environment, right? Do you agree on that? Disagree on that? Yes, no? Important for maintaining trophic cascade? Yes, correct. Yeah. So when you are removing an apex predator such as a tiger from an environment, it becomes really difficult to control the prey species, right? So that becomes a very important thing. So what can be done is you can monitor populations of apex predators or keystone species that are very important for an ecosystem, and then by monitoring those apex species or keystone species in an ecosystem, you get an idea of the ecosystem health, right? Like Afia mentioned, it's important for maintaining the trophic cascade. When there were no wolves in Yellowstone National Park, there was an surfeit of deer or other grazing animals that just managed to completely graze off all the vegetation, right? So there was a huge imbalance, and you need to know what are the right number of individuals to maintain in an ecosystem, so such that a balance is maintained, right? So there are various factors that can affect population density in animals. I think mostly everybody has mentioned everything. There can be a lot of factors that lead to loss of organisms. Can be due to some demographic stochasticity, which is basically variation in birth rate, death rate, or differences in the sex ratio, and also there can be other environmental factors. Does everybody understand what does the word stochasticity mean? Is there anyone who would like to explain, or would you like me to explain? Anyone would like to take a guess as to what is stochasticity?
It is a random probability.
Right, exactly, right? So any stochastic event is a random event that happens, and demographic stochasticity or environmental stochasticity is just any random event that can cause like some fluctuation in the population density or in some environmental effect, etc., that can result in numbers of organisms in any population fluctuating a lot, right? Alright, so the dash effect describes a scenario in which populations at low numbers are affected by a positive relationship between population growth rate and density, which increases their likelihood of extinction. Does anyone know or has heard of this particular effect? Anybody? So this is named after a scientist. Can't see if anyone's using the chat. Yeah. Any ideas?
Alley effect?
Yeah, correct. The alley effect. Could you tell me something more about the alley effect, or an example or something? Anyone? Anyone knows what an alley effect is? So, as the name itself, as the definition itself suggests, it is a density dependent effect, right? So, as the density increases, there is a increase in the population density also, right? So individuals are able to, for example, if the density, like the most common example is usually seen in guppies. Guppies are like small fish that are found in aquariums and fish tanks. So with the increase in the population density of guppies or adult guppies, they are likely to produce more guppies, right? So that's a density dependent effect that happens. And this also leads to ultimately competition, where there are too many individuals per unit space and they compete for the same resources. So the alley effect is basically a positive association between average individual fitness and population time over some fixed interval, right? So that's the alley effect. Can someone tell me what does sampling intensity mean? Any idea? So we're slowly moved into sampling to figure out population density. Can someone tell me what a sample is?
Maybe a small portion from a large population.
Yeah. So if you say that you want to measure the population density of let's say pigeons in India. Everyone has seen a pigeon, right? Pigeon bird. If you were to measure like the population density of pigeons in India, how would you do this? It's literally found everywhere, right? You can't really go to every single corner of a city or a rural outskirts to look for where pigeons are, right? So you need to do your sampling in such a way, which is basically you look at small units of the entire population and then extrapolate your results to estimating a population density of a larger area, right? So that is called a sampling unit. And number of units in the sample is divided by the number of units in the population is the sampling intensity, correct. Yeah, so the total number of probable units that you have, as Afia mentioned, is your population size, And then if you sample every 4 individuals in the entire population, your sampling intensity is 4 divided by like the total number, right? So that is sampling intensity. What does this image alongside represent, and in what cases are such techniques used? Everyone can see the image alongside. What does that look like?
Taking samples.
Taking samples. Any ideas as to what this method could be?
Cardboard sampling.
Carbon sampling. No, not really. Okay, leave about what they are doing. Yeah, it's a quadrat sampling. Yeah. So what exactly is quadrat sampling? Afia, would you like to explain? Anybody? Would you like to explain what's quadrat sampling?
Dividing the whole plot into quadrants, different quadrants, and taking out the samples from that particular coordinate.
Right. So it's a method of subsampling, right? You need to take a small area. You take a quadrat, which is a fixed unit space. This quadrat is basically nothing but a square. In this case, probably it's made out of some pipes or a frame. So you just take something of a constant quadrat measurement and you place it anywhere if you are doing some kind of sampling in that framework, and you count the number of individuals in one quadrat. You count the number of individuals irrespective of the species. You categorize them according to species and you count their numbers. So that's what you do in quadrat sampling. And in what cases are such techniques used? Quadrat sampling. You can't use can you use quadrat sampling to measure honeybees? Okay, after says pitfall. But can you use quadrat transects to study something like honeybees?
We cannot.
You can't, right? Why can't you? Because they move, right? And they're not going to sit in your quadrat frame. There's too much of movement in and out. You can't use that. You have to use it for something that is stationary or that remains fixed. And also, you have to use it for a particular organism that is distributed over a very large area, right? You can't physically go and monitor, let's say for example, marine crustaceans in an intertidal zone when you don't have, without quadrats, right? That's the most common way of intertidal zone population estimation of organisms in that area, right? It's to use quadrat sampling. So they are used for individuals that are mostly fixed and that are distributed over a large area. So in a heterogeneous population, the population is divided into subpopulations which are internally homogeneous. This particular type of sampling technique is known as random sampling, systematic sampling, stratified sampling, or multi-stage sampling.
Random sampling.
Okay, so one word for random sampling. What do the others feel? What do the others feel? What type of sampling is comprised? Anybody? Any guesses? No? No guesses? Stratified. Okay. One person for stratified. Sorry, I don't know your name. What else? Any other guesses? No other guesses? Just two people? Do you know or not know? If you don't know, please let me know. It's okay not to know. Yeah, just use the chat box or yeah. Let me know if you don't understand. Okay, so Afia says she doesn't know. That's okay. Alright, so actually the correct answer indeed is stratified sampling. Could the person who said stratified sampling maybe like to explain a little bit more with the help of an example or something? N? Yes, no? Okay, it would also be great if people could rename themselves with names. I see one succulent thorns also in this list. Yeah, it's just easier to, engage and have some interactions. Alright, so if no one knows or would like to discuss what is stratified sampling, I can give you an example. So basically, let's say you have an environment like the Shola Sky Islands. I don't know if any of you are familiar with the Shola Sky Islands. I can just show you an image. So this is the landscape in which I work and or our entire lab works, and these are high elevation cloud mountain sites in the western Ghats of India. So I don't know if you can see this page. Right, so can everybody see this landscape, which consists of like light green, basically these are grasslands, and these dark green patches are Shola forests. Is this visible? Alright, so so if you were to sample any vegetation in this area, if you did random sampling, there is, and you were supposed to map, like, let's say, vegetation of the Shola Sky Islands. If you were to do random sampling in such a case, if you were to randomly sample 100 points, what are the most likely points you are going to sample? In this case, let's just take for example this image. So the light green are grasslands and the dark green are forests. Which habitat are you most likely to sample? Would it be grasslands or forests? If you were to randomly sample, if I were to close my eyes and throw and point my finger to some, like point my finger 6 times on this photo, what area am I likely to point most towards? Excluding the, let's say, the sky, just if I'm focusing on the lower half. Which area am I most likely to point towards? Is it going to be forest or grassland? There are just 2 choices here. Put in your answers. Okay, Afia says dark green. What about the others? Forest, okay. So the forest are the dark green patches and the light green patches are grassland. 2 people say forest. What about the others? I'm gonna wait for a couple of more replies. Nobody? But in grassland also, correct. There is vegetation in both grassland and forest. This is the Shola ecosystem. If I were to sample vegetation in the Shola ecosystem, including grassland and forest, and if I chose to sample via random sampling, this is a huge area, right? I can't physically walk in all these areas to sample. If I were to randomly sample, 10 points and I put my finger on any 10 random points in this landscape, is it more likely to be a forest or is it more likely to be a grassland is the question. And 2 people say forest. What about the others? What do you feel? Am I most likely to sample in a grassland or a forest? Come on, just pick something. Okay, forest. Others? Come on, we need to have some more interactions. Who has not put in their things? Uzma, well, you have succulent thorns, Prashanta, N, Mrinal, Amrita. What do you all feel? Okay, let's break it down a little more. Which is a larger area that is covered out here? Is it forest or grassland? In this image, which type of habitat covers a larger area? Is it forest or is it grassland? This is an easy question. Grassland, correct, right? You can see this. You can see that the grassland is covering a larger area than the forest. So, yes, correct, grassland. So now if I were to randomly pick 10 points, are they more likely to be from grassland or forest? Keeping this information in mind, what do you feel? Okay, Renal says grassland. Grassland. Architram, what do you feel? Do you feel grassland or would you like to stick to forest?
Now I think this is for grassland.
Okay, so of the 3 people that replied, after getting this piece of information, all of you feel that it is grassland, right? Because whichever is the majority of the area, just by a matter of probability, you are more likely to randomly sample a grassland. Like your odds of sampling a grassland, 10 grasslands is going to be much higher than sampling 10 forests, right? Now, if I'm supposed to do a vegetation assessment of the Shola ecosystem, I should be covering forests as well as grasslands, right? But if I do a random sampling, I will not get to sample forests. So, I need to do a sampling in such a way that some of my plots are forests and some of my plots are grassland, right? So that brings us to a type of sampling which is known as stratified sampling, right? Where your population is divided into subpopulations which are internally homogeneous. So I will have a plot that will be grassland that will be sampled and a plot that will be forest that will be sampled, right? So I hope this is clear to everyone. I hope this makes sense. So that is stratified sampling for you. We can go to the next question. Alright, awesome. Okay, so take out your pen and papers or do it in Excel. So tell me, you need to, okay, I'm going to read this out. So you are on a small island, and what you are a scientist and you are trying to find out the population size of a particular butterfly species, right? The technique you will be using is a dash method, and using this method you catch 90 butterflies on day 1 and paint a white cross on their wing. On day 7 you return and you caught, sorry for the typo, 80 butterflies. This includes 16 with previously marked crosses, right? Please ignore that typo again. This includes 16 with previously marked crosses. Given the information provided, the population size of the species is, 2 people say this is the mark and recapture method. That is absolutely correct. So can you please calculate what is the population size of this species? I'll give you about a couple of minutes to calculate. Let me know what is the right answer. Okay, one person with 450. Prashanta says 450. What about others? You can calculate and let me know. I'm gonna wait for a couple of more replies before we proceed. Okay. Okay, so I have a couple of answers for 450. Could you tell me the formula for mark and recapture that you're using? You can unmute or type it out. Okay, so I actually have the formula written out here. So basically, the mark and recapture formula includes animals marked and released in the first instance is equal to the number of marked individuals caught in the second instance and the total number of individuals caught in the second instance. So your animals that were caught and marked in the first instance were 90 butterflies. And when you return and you catch 80 butterflies, that's the total number of individuals caught in the second phase, which is 80 butterflies. 16 of them were marked. So those are the marked individuals caught at the second instance. So just if you rearrange the variables on the left hand side and right hand side, you get 90 into 60 divided by 16, which is equal to 450. So now tell me, what are the drawbacks of using this method? Can you think of any drawbacks as to why we should not be using mark and recapture, or why, in what instances mark and recapture might fail? Yeah, that is one of a real big problem, right? If we
the individual dies during the, during the count, during the first and second recapture,
right,
then this will be a failure.
Yeah, right. So you won't be able to estimate populations correctly if you lose individuals, especially if you lose out mark individuals due to some mortality, or they are not caught, or something. It's going to be a little difficult, right? So you really won't be able to make out mortality in marked and unmarked individuals. As Afia says, the marking can easily just come off, and then the next time you catch this individual, you will think it's an unmarked individual and you will be marking it again. So that's going to be some overestimation. And what is another assumption of mark and recapture?
We're assuming that the population is static throughout the entire period in which we're measuring. Is that right?
No, actually not really static, because we are able. So what is an assumption of the mark and recapture is basically that whatever unmarked individuals that you are catching the second time are new additions to the population, right? So it's like a proportion. But there is something else that we assume. Amrita, I think you are going into probably something like Hardy-Weinberg, which is not the case in this. So you are, if the population size is increasing, no, it's, well, okay, sure, closed population, because, well, there are no infinite populations on Earth. In terms of assumptions, but there is another important another important factor. It might be a little difficult to visualize. Okay, let me give you an example of something that I do. Or our lab does. What we do is we sample birds in populations, and what we do is we put these specialized mist nets in forests to trap birds, right? So these are like 12-meter nets which are put in between two poles, and we put these in particular areas at particular times in the forest, and these birds come and are trapped in these nets. Now when we go the next year to the same location, for example, we will put up this nets in exactly the same place for the same duration of time, and then look at the number of individuals that are caught and are not, right? So look at it in terms of like the equation itself. There are cases where I catch a lot of marked individuals. What does that mean? When I catch a lot of marked individuals in the second instance, what will happen to the estimation of my population size? Just look at the equation and tell me. If the number of marked individuals that I caught in my second sampling is large, what does that mean in terms of population size?
Population is very less in the nature.
Population is very less. Okay, let's look at the equation. Is it directly proportionate or inversely proportionate? Estimated population size and marked individuals caught in the second instance. Everybody knows directly proportionate, inversely proportionate. If something increases, if variable 1 increases, variable 2 increases, that is called directly proportionate. But if variable 1 increases and variable 2 decreases, it's called inversely proportionate. So what does this equation show? Inversely with recapture. Okay, so if I recapture a large number of marked individuals at the second time, what is likely to be estimated for my population size? Is it going to be high or low? Right. I can't remember. I think it was Architam who replied. Architam, what did you say? Okay, right. So, this is yeah, you're right. So I hope everybody understood this. If your number of marked individuals that you catch are very high, your overall population density estimation goes low, right? And this happens when certain individuals have a higher affinity to get caught, right? There are some animals that keep on using those paths, and though there are more individuals that have been recruited into the population, it won't be apparent because you will be catching roughly the same marked individuals the second time you are capturing, right? So an assumption of this mark and recapture framework is that marked and unmarked individuals are caught randomly. But in this case that I described right now, do you think it is random or not random?
Random.
In the case that I mentioned with the birds, some birds having an affinity to go to the net, is it random or not random? Correct, right? It's set in the same place and I'm catching the same individuals. It's not random, right? Because if I were to catch, if this was random sampling, for example, what do you think would be the probability of catching marked and unmarked individuals? you should have equal probability, right? You could either have a marked individual or you could have an unmarked individual. But if I'm saying that the probability of you to catch a marked individual is higher, it makes it not random, right? You have a higher probability of catching a marked individual as compared to an unmarked individual, so that makes it non-random. So this is one of the assumptions of mark and recapture, and one has to be careful when you do these kind of studies, because you can account for all of this in any particular way as well, but this is something that should be kept in mind. Was this clear? Or do you need some more? Does anyone want me to explain this again? Alright, I hope this is clear to everyone. So let's move to the next question. Okay, again little bit of calculations. So given this is a transect, okay, I have put a transect like a square transect grid on a marine coastal area, and these are the number of individuals that I count in the sub-quadrats of this large quadrat. I know it doesn't look like a square, but let's say it's a rectangular quadrat divided into 9 subpopulations. Can you calculate the mean, standard deviation, standard error and coefficient of variation? You can again take a couple of minutes to do this. Meanwhile, can somebody explain to me what is a mean?
Average.
Average, right? So how would you calculate an average? It's very easy. But tell me. Everyone knows how to calculate an average. You can even put this in Excel and tell me the answer. Tell me what is the average, what is the average number of individuals that you are getting. Anybody with any results? Just use Excel, use your phone, anything. Nobody has any answers for average? Well, mark and recapture seem to be calculated faster. Alright, one answer, 4.44. Others, do you agree with Afia? Anybody else? Nobody else? Can someone tell me what is the standard deviation? Standard error. Does everybody know what is the formula of standard deviation? So an important thing about doing ecology is also doing a lot of statistics, because ecology and statistics go hand in hand, and these are some of the basic stats that you might have learned like in school, but it's good to remember. Any answers? Nobody? Or is this too basic? If this is too basic, then we can go to the next question. I don't mind. Does someone need any explanation to this? You can thumbs up. If not, then we can move ahead. Okay, you're the only one who is actually answering, but okay. Alright, so your mean is basically an average, right? Your nothing but a total of number of individuals caught in every square divided by the total number of squares, right? That is your average number of individuals per quadrat, which is, as Afia has said, 4.44. Now your standard deviation is basically nothing but x minus x bar divided by n, right? So x in this case is every observation, which is your 1, 3, 9, 5, 8, 6, 1, 6, 1, and what you have to do is subtract each observation by the mean, which is what you calculated, and summate that and divide it by the total number of observations. What are the total number of observations here? 9. Correct. So you have a summation of x minus x bar divided by 9. So you have basically 1 minus 4.44 plus 3 minus 4.44 plus 9 minus 4.44, so on and so forth, divided by 9. That's going to give you the standard deviation, which is basically how much variance is there that is captured by your mean, right? What is the deviation from the mean in your population? That is your standard deviation. Then you have a standard error. So standard error is also gives you a similar statistic to a standard deviation, but it gives it to you with respect to the entire population, right? So standard deviation measures how much observations particularly vary from each other, while standard error looks at how accurate the mean of a sample data is compared to the true mean of the population. So you must always remember, when you're sampling, as we discussed in the initial part, small groups from a large population, right? But you need to see how much is the variation from the population mean, and for that you compute a standard error, and the formula of standard error is nothing but your standard deviation divided by the square root of the sample size. So in this case, the standard error that you calculated in B divided by square root of 9, right? That gives you the standard error. Now what does the coefficient of variation do? The coefficient of variation computes the differences between the standard error and the mean, right? And for this, what the coefficient of variation is calculated by dividing the standard error, dividing the standard deviation by the population mean, and you multiply that by 100, right? So that gives you a ratio. Okay, so I hope all this is clear to everyone, and please make sure you go through the lectures. I think this was a part of lecture 14 where all these calculations are given, so make sure you read through them and do understand what these calculation metrics are. Alright, so let's get into something like research design or something. So you are assessing the biodiversity in a forest. You are a scientist, you are assessing the biodiversity in a rainforest. What would be the possible ways in which you could conduct surveys with minimum human intervention? Could you think of some ways? Don't worry about being right or wrong, we can discuss.
Using IoT technologies and AI.
Using what technologies?
IoT.
What is that?
Internet of Things, using machine learning and IoT sensors.
Okay, could you give me some examples?
For example, assessing the soil moisture in real time using soil moisture sensors.
Okay. Okay. So, if you were to, let's be more specific. I like your idea. You can build on that. But if we were to focus on the biotic factors, not the abiotic factors, because biodiversity is basically the biotic factors, right? So, yeah. Prashanta says camera traps. That is one way. What are the other ways? So camera trap is, Prashanta, would you like to explain how camera traps work and what camera traps do?
Yes, the area is first of all divided into multiple grids.
Okay. Yeah.
And then camera has to be installed.
Okay.
These cameras are motion sensors.
Correct.
When something is passed, then usually it is placed front to front, so when something is moved in between them, the photograph is taken.
Okay.
And both sides of the animal is captured.
Right.
This is particularly very useful in tiger census programs.
Okay. Yeah. So yeah, thanks Prashanta for the explanation. So basically, as he explained correctly, camera traps are used to get like a visual identity of whatever organism is passing. If you're interested in counting the number of individuals of tigers that are present in a particular locality, you can put up camera traps, and tigers use certain trails or paths or usually roads and well established paths. So if you position your camera trap in such a way that you know you are going to get frequent traffic of animals out there, it will be triggered, as he mentioned, by a motion sensor. There are two camera traps usually that are present opposite to each other, so that you can catch both sides, and as the individual passes and crosses, a sensor is triggered and you have an image of the animal. So camera traps is one way. What is another way in which you can monitor biodiversity or assess biodiversity without much human intervention? So why are camera traps having less human intervention? Because you can just go and deploy them in the field and then collect them after several days and just assess the data, whatever has gone into it. Collect the photos and then analyze it. What are other ways? This is interesting. I didn't think anyone would mention this, but yeah, sure. Scat sampling. That's something you can do. Yes, succulent thorns. you have a question? Okay, so scat sampling. Scat sampling probably is one way to conduct surveys, but to sample scat, you still would need people, right? Because people would have to be walking in the forest along trails and looking for scat. That is definitely a way in which populations can be monitored. Great example. But we are looking for something which has very minimal human interventions. You don't need humans to be walking around. Any ideas? Okay, let me give you a clue. Camera trap is some way to get visual information. What are the other ways in which you can get other types of information? What are the other types of information that you can get? So one is visual information. Amrita mentioned scat. What else? How else can you? Yes, correct, right? Sound. You have sound. What is study of sound sound used to study organisms called? What is that study called? It's known as bioacoustics, right? You can use bioacoustic technology. So just like camera traps, you can use automated recorders. So what are these automated recorders? They are programmable instruments where, just like camera traps, you can schedule them to capture calls at certain points of time in the day, and you can get like a schedule that is fixed into it, programmed into it, and all you have to do is put it on a tree. It will have a microphone that will collect sound, and it will collect sound for as long as you have programmed it for, and the battery lasts. So this using bioacoustics or passive acoustic monitoring is another way in which you can assess biodiversity without minimum human intervention, right? And then you will have a lot of acoustic data that you accumulate, and you can create different detection algorithms and use AI to create detectors that can detect different species across different taxa. You can study frogs, insects, birds, mammals, because everything vocalizes, right? Everything makes some sound at some point of time. So you won't get to know how many crickets are present or how many frogs are present if you're doing something like camera trapping, right? You'll only find larger animals. So acoustics is another way to understand the biodiversity that is present in an area, and especially a rainforest, because it's a highly active acoustic space. Alright, so the figure alongside denotes what kind of trap? Any guesses? Pitfall trap, sticky trap, bowl trap, pan trap. Okay. Any other guesses? Anybody? What does it not look like? Okay. one more for hole trap. Okay, okay, cool. It does not look like a pitfall trap, that's clear. Okay, so actually the term for this kind of trap is known as a pan trap. Anybody knows what are the constituents of a pan trap? Yeah, pan trap. What do you have in a pan trap? How do you trap insects? Okay, what is a pan trap used for? Insects, correct. What is usually present in a pan trap? Insects, okay. What is present inside the pan trap? What is any idea as to how insects are trapped in that? Is it sugar solution? What is it? Correct. Yes, it is an agent it has some kind of substance that kills and traps the insects. Preservative, yes, and it's mainly usually a soapy solution, right? It's just a soapy solution. And what is interesting about this figure? Why are there multiple colors of plates used here or pans used here? Why are they not all the same color? Is it like something random, or does it have any significance? Any ideas? Is the color choice just correct, right? See, different insects have an affinity to be attracted to different colors, right? So that's why out here, to capture like the entire range, they have put 3 colors, because some insects that have an affinity to which don't have an affinity to yellow might be having an affinity to blue or white, right? So this is to capture a wide range of insects. Alright, so we have an image out here. The following figures show 4 possible population demographics for age structure. Okay, which of the following graphs best represents a country with a fast growing population? So can someone first tell me what is shown in this image? Any ideas? You have seen these graphs. Age pyramid, okay. Any guesses as to which figure A, B, C or D could represent a fast growing population? Okay, one answer for A. Others, what do you feel? Right, so you have age on the y-axis, right? and you have a percentage or usually a ratio of males and females on the left and right side. So tell me, which of the following graphs best represents a country with a fast growing population? What do you expect in a fast growing population? High natality, correct. Now, which of these figures show high natality as compared to the others? Which of the figures show a highest degree of natality? Okay, so right, so high natality is seen in A and B. Now, if you look within both of these as compared to the other population structures that are given within a graph, which graph do you feel shows the highest natality? Any guesses? Now we have shortlisted two graphs. Okay. Actually, so this is probably like an illusion, but the width of the width of the younger population, like the light blue in both the graphs, is exactly the same. The bar. So if you look at the natality of the number of individuals that are the youngest, like let's not even say 0 to 25, let's say 0 to 5 as a proportion of the other older age groups, which may be an exponential graph line, which population seems to be having a much higher natality as compared to the, let's say, mid-range population between 25 to 65. Let's go to the slide. So here you can see the light blue on graph A and graph B. The number of individuals in light blue as a proportion of number of individuals in this darker blue as compared to the number of individuals in light blue here to the number of individuals in dark blue here, which would be greater? Just visually, could you tell me which will have a higher proportion of younger individuals? Right. So Prashanta says A has the highest natality, right? Because if you divide the number of light blue by dark blue, you are going to see a much higher ratio for A, right? So that means that the birth rate is higher in A as compared to B. B is also a growing population, but A is a faster growing population because the number of individuals between 25 to 65 is decreasing at a much higher rate as compared to 25 to 65 in graph B, right? And the death rate is even more high for A as compared to B. So yes, both are expanding populations, but the faster growing population is seen in graph A. What about C and D? What do you feel about that? What kind of population is shown in C? C is stable. Okay. Everybody says C is stable. So could you give me a few characteristics of the stable population? B. I'm asking about the figure C. What do you feel are the factors that contribute to a stable population? Declining birth rate. Okay. What else? As compared to A and B, what is also different in C? You have a lower death rate, right? Yeah, so you have a lower death rate. And what about D? Prashanta says D is declining. What are the characteristics of a D population? You have a large number of individuals in the higher age group, which means that the death rate is pretty low. What's happening to the birth rate? Birth rate is also low, right? So this is basically nothing but a declining or a contracting population. So I hope these graphs are clear to all of you. Low reproductive population. Actually, the reproductive population is high because the reproductive population between 25 to 65 is high. But people are living for much longer, and there is something that is happening to the birth rate. So the number of individuals that are born are very less, but the number of individuals that are reproductive in the reproductive age group is still the same or high, right? So basically people live for longer, but they are not very reproductively successful. Okay, so I hope this is all clear to everyone. And does anyone have any other questions? If not, then we can close the session and we can meet next week. Anybody? Any questions?
No, ma'am.
Alright. Alright, thanks for joining in today, and see you all next week, and hopefully we have more people. And thank you for everyone who participated and very actively interacted for the session as well. Thanks for joining in. Yeah. Alright, bye.